So welcome everybody to our West Hour, which is webinars empowering science and technology for those that don't understand what that stands for. Our very own Dr. Ian Villamagna, the field application scientist from the Northwest, will be presenting on a topic that we really can't ever get too much information on: overcoming key challenges in vaccine development. So thank you for attending. Just a quick reminder that Ian is part of the Northwest territory, where Angel Galarza is the regional sales director and Ian is the field application scientist. I'm Kim Killian. I'm the regional sales manager for the Southwest territory, located in San Diego, and I work very closely with my field application scientist that everyone knows, Viet Nguyen, and he is located in Irvine. And if you're new and you'd like to catch up on all of our other webinars, which we have a webinar the last Wednesday of every month, you can check out our YouTube playlist, and you can find it again under West Hour. And if you have to leave early, this will be recorded and you can go back and listen to it. So I believe that's everything. I'm going to, again, welcome Ian and thank you for presenting today.
Great. Yeah, thanks, Kim. And thanks, everyone, for making it today to chat again, it feels like, about vaccine development, which is obviously really important to a lot of us right now. And we were hoping to give a really good overview of some of the solutions that PNI offers to assist with that vaccine development overall. So just to start for today, like we always like to start with our vision. Our vision at Precision NanoSystems is to accelerate the creation of transformative medicine that significantly impacts human well-being. And obviously, we believe that within the past year and a half or so now, vaccines have become a really key component of that vision, and ensuring that we can really accelerate the creation of those vaccines, which are, as we've seen, truly life-changing at this point. So, with vaccines, I think within the past year and a half, we both at Precision NanoSystems and we as a collective field have really learned a lot about vaccine development in general, and have really been able to grow and expand on what we knew about vaccines and kind of change the way we think about them in general. I think overall, what we kind of can take back and look at right now is that we know a rapid response is really critical to emerging issues such as viruses or emerging variants that are happening. And we do know now very commonly that variants can and will occur. So we're seeing this right now with the Delta variant making its rounds throughout the world, really. And we have to be ready to respond to not only the original virus, but also variants as well. The other thing I think that we've really learned within this past year or so is that production capacity is still a bottleneck. And getting up to production for vaccines is still a challenge. But I think we've been able to take great strides and make a significant amount of vaccine. Of course, I say that as someone in the United States where we had great access to vaccines, but knowing full and well that the rollout in the rest of the world hasn't been as rapid. And it's something that I think we need to continue to grow on and build on in the future. And the reason for that is because everyone really needs protection. In order for things like pandemics to come to an end, we really need to get as many people vaccinated, not only in North America, but around the world. And so everyone needs protection, and we need that production capacity to not bottleneck at any stage. So if we look at current vaccine technologies, this is actually a figure that's taken out of a recent review publication by Anna Blackney at the University of British Columbia, and working with a couple of individuals from our team at Precision NanoSystems. There's obviously a number of different types of vaccines right now. And, I think probably most commonly associated now with COVID vaccines would be the nucleic acid vaccines that we've seen being used, both through synthetic carriers and viral vectored. And I think what we've seen here with these nucleic acid vaccines, which you're looking at on the right-hand side of this figure, is that overall, they have been shown now to be safe and very effective. And they are easy to manufacture, relatively speaking, especially compared to more traditional vaccines or something like a live attenuated vaccine or a whole inactivated virus vaccine. There are still some challenges that occur with them. So with something like a synthetic carrier or a lipid nanoparticle nucleic acid vaccine, we still look for things like limited immunogenicity and how we increase that immunogenicity to really increase the effectiveness of that vaccine. And with viral vector vaccines using nucleic acids, we still do see that they can be difficult to manufacture due to a number of reasons, really. So I think that we've made these great strides, and nucleic acid vaccines definitely appear to be the future. But I think there are still a lot of strides that we can take to grow on that. Now, obviously, for the purpose of this talk today, and for a lot of the talks you'll hear from Precision Nanosystems, we will focus our efforts on RNA vaccines. And so, I think RNA vaccines really showed quite effectively that we were able to have this very rapid response to an emerging threat. So it can be kind of hard to think back and remember the start of COVID-19 and all of that. But essentially, in early January, Moderna did get the sequence for the genome of COVID-19, or for SARS-CoV-2, I should say. And in between the time they got that sequence and they were able to go to manufacture for clinical trials, it took them just 45 days. And so essentially what they went through was sequencing, looking through an antigen library and selecting an antigen that they could encode for, manufacturing the RNA, encapsulating that, and then going through final processes like fill finish to get to administration. And so with an RNA vaccine, it took just 66 days for the first trial participants to be vaccinated in a phase 1 clinical trial, which was a much more rapid time frame than I think we had ever seen before or ever really imagined would happen. Now, what I think is really interesting about this timeline and our understanding of how quickly it went is that I don't think it was all that shocking to some people in the field, because Moderna had been working on things like this for quite a while. And so this paper here actually came from 2017, and it came out of Moderna. And in this publication, they were actually looking at, excuse me, RNA vaccines against influenza viruses, 2 different influenza viruses. And essentially what they were looking to do is create a system with an RNA vaccine that could be prepared to respond to a pandemic. They knew that pandemic outbreaks would spread very quickly and that the traditional vaccine approach was quite slow. They had estimated in this publication that they were looking at about 5 to 6 months of a time frame to get from essentially inception, or when the pandemic started, into having a vaccine to go into trials. And so what they began to look at were really the basis of these RNA vaccines at this type of scale. So, how do you stabilize and deliver that RNA? Is it a safe and effective method to use? And they did this with that influenza model. And through this publication, I think, a lot of what they show here is a lot of preclinical data coming from different animal models, and also some early human data as well. What you can see here is in the top row across the top is looking at a mice survival rate when these vaccines were given. And it was pretty clear from the beginning that the percent survival of these animals was quite high when they were vaccinated against the flu strain that was in question here. At the 10 microgram and 2 microgram dose, they had near, if not 100% survival, and still much higher survival at 10 days post-infection, even with a 0.4 microgram dose. They did challenge these animals 3 times. So you're looking across the top row here at a 7-day challenge, a 21-day challenge, and an 84-day challenge. And across the board, it did appear that the survival was quite good. So giving them an indication that these RNA vaccines were going to be quite effective overall, safe and tolerated in an animal model. And then along the bottom, what they were looking at was the actual titers for different flu strains, so H10 and 8. And they looked at that in both mice, monkeys, and humans as well. And they were able to see those titers quite efficiently at a number of different days post-immunization. So showing that, not only was the survival there in the mouse model, but that they were very effective overall at inducing protection, if you will. And so I think, this study was and still is a really great one to touch on. And it's one that I go back to quite often, especially when we're working with groups that are just getting into the vaccine space. I think it is a really great overview of vaccines in general and kind of RNA vaccines and how they work, where they came from. But I do think that, the understanding of publications like this kind of showed, even before COVID-19, that this was something that was doable and was something that could very well change the progression of a pandemic. So obviously now we've showed, an RNA vaccine and we've seen them, a lot of us have probably received an RNA vaccine, but what does it actually take to get to that point? What does it actually take to make these and produce them? What we work with at Precision Nanosystems, we like to call our Vaccine Toolkit. So basically a suite of technological platforms that really enable the rapid development of vaccine development. And what that Vaccine Toolkit really comprises itself of are, I would say, 3 different platform technologies, which you're looking at in the center of the screen here. So essentially, where a lot of our customers come to us, they have a disease target that they've identified. In some cases, they have RNA prepared. In some cases, they don't. And where the Vaccine Toolkit really comes into play is that we first and foremost have a genetic payload platform. So vectors that can encode the antigen for RNA replication. We also have a GenVoy delivery platform, which you're looking at in the center there. So this is a lipid nanoparticle-based platform that is quite versatile and can be used to encapsulate a number of different nucleic acids. And then obviously, for anyone that's familiar with Precision Nanosystems, we have our manufacturing platform, the NanoAssemblr Platform, which is there to essentially really effectively, really rapidly encapsulate that RNA into a lipid nanoparticle and really allow for it to be delivered in a vaccine product. Along with that toolkit, we have really end-to-end development support. So drug development expertise is really one of the things that we have here at Precision Nanosystems across the board. And whether that comes in the way of any of our field application scientists at the early stage of development or anyone back in Vancouver with all of our drug development expertise there, there is almost in every instance someone that can help answer any question that you have in that development process. So we'll kind of touch on each of these different components a little bit more here, starting with RNA technology. And I think one of the things that that we started to see about probably towards the latter half of 2020 was the idea that we can have RNA vaccines with very different types of RNA. And I think for a lot of us in the field, it probably didn't come as an outward shock. We're already well aware that we could use things like siRNA versus mRNA and things of that nature. But even within the mRNA category, there are subcategories, and all of these subcategories are being used for vaccine development. So you have mRNAs such as unmodified mRNA, in which they've enhanced the stability of the RNA, but they don't do any major base modifications to it. And companies like CureVac are working on that for vaccine development. Most commonly associated with these mRNA vaccines would be base modified mRNA. So here they're essentially changing some of the bases in that RNA to change the immune response that they're getting from them. This is what Moderna and BioNTech Pfizer were doing and did with their vaccines, was a base modified mRNA approach. And then I would say more recently, what we've kind of seen come out is this idea of using self-amplifying RNA. So an RNA that can be delivered in a much lower dose, but can act as its own adjuvant. So it can actually replicate and make copies of that RNA to really amplify the effect that it has. This is what Precision NanoSystems is working on in our current vaccine program. A couple other of our collaborators are working on it as well. And companies like Gritstone as well now have shown that they're using a self-amplifying RNA model to essentially create COVID vaccines. Really, when we think about self-amplifying RNA and why it becomes so important, I think what it really comes down to is that ability for it to amplify itself. And so, from a really basic level of understanding here, what this would essentially look like is you would deliver the self-amplifying RNA, and before it actually encodes and makes those proteins that it's encoding for, it actually makes more copies of that RNA. And so what this enables you to do is essentially deliver much lower quantity of RNA, but still potentially get that same, if not better, immune response and actual protection from that vaccine. And this can really come down to two different factors, I would say, where it becomes really important. The first being with a lower amount of RNA, there's potential for having lower side effects, although I would say we're still kind of looking at that overall with different trials. But the other area that I think this really becomes important in is just from pure production capabilities. If you need only a tenth of the amount of RNA to be delivered, you don't have to make as much RNA from the start. And so going back to the beginning where we talked about production concerns and bottlenecks and that need for everyone to be vaccinated, I think the overwhelming idea at this point is that if we can have a vaccine with as little RNA as possible, we're in a good space because we have the ability to make that RNA at a much more rapid pace, encapsulate it, and get that vaccine out to people at a much more rapid pace. So the next platform that we'll kind of touch on here is our GenVoy delivery platform. And so GenVoy is a lipid mix platform that we have at Precision NanoSystems. It is an ionizable lipid mix, so quite similar to what you have seen or heard of with the Moderna and BioNTech Pfizer vaccines, although slightly different. It is a proprietary lipid mix to Precision NanoSystems. And what it really enables is the development of really robust lipid nanoparticles that can really efficiently encapsulate RNA and protect it for that administration. So the components of the delivery platform and these lipid nanoparticles are first and foremost an ionizable lipid, which you see in light blue here, which is really there for the interaction with the nucleic acid. Other components that are within these nanoparticles would be things like cholesterol and helper lipids for stability and then uptake purposes, a stabilizing lipid around the outside of that particle to keep it from aggregating in solution, and then obviously a nucleic acid payload. Now, these particles that are made with this platform are extremely versatile. We have been able to see with this platform that we can encapsulate really any number of cargos, which we'll talk about a little bit here. And the uptake is quite efficient because of the cholesterol in these particles. These particles undergo uptake through an ApoE mechanism into cells, which typically works quite efficiently for vaccine approaches. In terms of, like I mentioned, GenVoy being able to encapsulate a variety of payloads, this is something that we've seen consistently across the board when using this lipid mix. And that comes both from us internally at Precision Nanosystems, but also from some of our collaborators as well who are using this in their programs. And so with that same lipid mix, what you're looking at here on the left-hand side is the size and the polydispersity of particles that were formed with 3 different payloads. So on the left-hand side, you're looking at self-amplifying RNA, which is quite large, so about 11,000 nucleotides. In the center, mRNA at 850, and then on the right, actually siRNA. So 3 very different sizes of RNA. And across the board, what we were able to see is that through the alteration of formulation parameters, such as the N-to-P ratio, so looking at the ratio of lipids to RNA and altering that based off the size of that RNA, we are able to formulate particles with all 3 of these payloads that are just over 50 nanometers in size and have polydispersities below 0.2. On the right-hand side of this figure, what you're looking at is the encapsulation efficiency as well. And so the encapsulation efficiency here was unaffected by the size of that payload. And so what you see with both or all of these payloads, the self-amplifying, the mRNA, and the siRNA, is that we are right above 90%, probably closer to 95% encapsulation efficiency. So the bulk of the RNA that's being added into the system is getting encapsulated and will be available for use in those particles. To kind of get an idea of how the actual GenVoy delivery platform has been used, we can look to some data from some of our collaborators. So this data was generated in collaboration with Robin Shattuck and Anna Blackney when she was still at Imperial College London. And like I mentioned, she's since moved to the University of British Columbia. But here what they were doing was taking an initial snapshot, if you will, of how these lipids compare and how these lipid nanoparticles compare to a more traditional polymeric-based delivery system that they had been using. So this was a polymer system in Payball that was really working off a charge-based system to essentially encapsulate, if you will, RNA and deliver it. And they wanted to compare that to the GenVoy delivery platform. And so what you're looking at in pink, orange, and green here are 3 different formulations of that GenVoy delivery platform. And here, what they were actually looking at was the hemagglutinin response. So looking at more of a flu model for mice administrations and for mice immunizations. And what they were able to see is a very strong IgG response in a number of different timeframes for all of the lipid nanoparticle-based vaccines, and much higher than they were seeing with their traditional polymeric system. In terms of efficiency and then safety for these animals, like you're seeing on the right here, by way of body weight measurements, what we were able to see across the board with the GenVoy delivery platform and with these lipids is that we did not see a large drop-off in body weight over 8 days. But when compared to the polymeric system, we did see an initial drop in body weight at around day 4 before it slightly recovered. And compared to a control in black, we did see a much better response to those lipid nanoparticles where we don't see that drop in body weight. These clinical lipids were also used, or one version of these clinical lipids, I should say, were also used for a SARS-CoV-2 self-amplifying RNA delivery. So at the onset of the pandemic last year, Imperial College and the collaborators that we were working with there kind of shifted gears a little bit and began working on this SARS-CoV-2 self-amplifying RNA delivery. And here what you can see is the IgG response to the SARS-CoV-2 specific RNA that was delivered. And so again, comparing it in black to that polymeric system, what we can see is that we do get a pretty strong IgG response at doses such as 1 microgram, and that it is a dose-dependent response overall. One thing that's interesting is that the traditional polymeric system that they were using in black is most comparable to the lipid nanoparticle formulation that you're seeing in pink here, even though the dose is much, much lower for that lipid nanoparticle-based system. Also interesting here on the very far right-hand side in that teal type color, what you're actually looking at is a 1 microgram dose, but rather than having an intramuscular injection, it was an intranasal injection. And it did show an IgG response, so giving us some indication that these types of encapsulations could be used in an intranasal fashion rather than just intramuscular as well. In addition to this, what we were able to see with these lipid nanoparticle approaches is at the higher level doses, which is where we would expect to start essentially looking at dosing. We were able to see here that we do induce a T cell response. And so I think, back in the latter half of 2020, one of the first times we talked about this publicly with this data, one of the big questions was, do these RNA vaccines induce that T cell response? And I think what we've been able to see here with these lipid nanoparticle RNA vaccines is that they do induce that T cell response in the mouse model. And I think we've seen data as well recently from the approved vaccines that they do induce a T cell response as well. So to give you kind of an idea of what a time frame looked like for Imperial College London, we started working with Imperial College London in December of 2019, so actually a fortuitous time. March 2020, they were doing the influenza challenges using that clinical lipid library, like I had mentioned in the previous slide. And in June of 2020, they shifted gears toward that SARS-CoV-2 model instead. By December of 2020, they had done a lot of the initial work for mouse immunogenicity, and they had moved into essentially tech transfer and looking at a manufacturing process. In March of 2021, or somewhere around there, they began engineering runs for tox and safety, and then produced a GMP batch in May of this year. And they are still set to start their phase 1/2 clinical trials in July of this year. And then finally, and I think really where that timeline is enabled in a great way is through the manufacturing platform, like I mentioned. So Precision NanoSystems was born off the NanoAssemblr platform in general. And so what the platform is really comprised of right now are 4 different instruments that enable the development of not only lipid nanoparticles, but nanoparticles in general. And they enable that at different stages of development. So starting on the left-hand side, we have the Spark, which is a rapid discovery tool. Makes really small volumes and is designed to rapidly encapsulate libraries of payload for in vitro use. The Ignite is where most of our customers actually meet us and begin their work. So this is really a formulation development tool for a number of different nanoparticle applications. It makes between 1 and 20 mils of nanoparticle formulation. So typically a really good spot for nanoparticle development, for formulation optimization, and very early stage animal studies, I would say. Since we always have to think about scaling up and that production need, especially with vaccines, that's where an instrument like the Blaze really comes into play. So the Blaze is really there to model scale up at a GMP level. So showing how these nanoparticle populations are going to respond to that scale and respond to that scale up. That's really what the Blaze is designed to be used for. So it makes between 20 milliliters and 10 liters of volume. So a very wide range there as well. and does produce material that can be used for those large-scale animal studies towards the end of development that are so crucial to the success of any program, but a vaccine program especially. And then finally, on the right-hand side, our GMP system is there to produce clinically relevant and clinical manufacture for phase 1-2 clinical trials, and has the ability to make really a wide range of volumes at extremely high flow rates. So really bringing that production to a level that is suitable for the needs that we have today. All of these systems really work on what we call next-gen microfluidics, which you're looking at on the right-hand side here. And what that really refers to is a method of mixing by which we can essentially combine our lipids and our RNA molecules in two different liquid forms, so ethanol and an aqueous buffer. And through that mixing technology, what we're able to do is really leverage the self-assembly process of these nanoparticle populations and form very consistent, very homogeneous nanoparticles. So a particle population that has a very low polydispersity and is very reproducible overall. And because our mixing technology allows for that self-assembly to occur, what that enables us to do is move this next-gen mixer through that platform that I just mentioned. And so what that means in terms of a vaccine or a therapeutic development project is that anything that is done at the Ignite level at 12 mils per minute can be scaled up to the Blaze, which runs at 60 or 90 mils per minute, and then can be run on the GMP system at 200 mils per minute. And so what that enables essentially here is a much more rapid movement through the development process with these lipid nanoparticle based approaches, whether they be vaccines or therapeutics. You're able to essentially move through and create these at a much more rapid rate than you would be otherwise. One more example here from Seronomics, which is a company that we work with quite frequently, and kind of a case study of how they ran forward with their production. Essentially, in October of 2018, they had a benchtop, so NanoAssemblr benchtop. That was the previous iteration of the Ignite. They really began working with lipid nanoparticles on the benchtop, I would say, in October, November of 2019. In June of 2019, they took purchase of a Blaze and they began formulation scale-up, essentially, looking at that, and then large-scale animal studies as well. By December of 2019, they had moved into that GMP system and were looking at engineering runs and tech transfer to a CMO. They did some engineering batch runs in early 2020, and then essentially handed it off for tech transfer later that year, and allowed for essentially the manufacture to commence. So a very rapid timeline overall, but I think this is a timeline that we see pretty consistently, and we've seen a few times now with some of our customers where with the right team in place, you are enabled to move quite quickly through this formulation development and therapeutic development program overall. And that's true for therapeutics or vaccines as well. The platform really does enable that rapid movement. And so I would definitely like to thank you all for kind of sticking with us and joining us here today. I know that I've seen some questions pop up here, which we'll definitely run through here at the end. One thing I just kind of like to close with, I think we talk about vaccines a lot with a lot of our customers, and typically, I think what we try to do is give examples of different types of publications where the NanoAssemblr platform was used, or something that we think would be a really good kind of indication for what can be done overall in this space. All of these publications here are ones that I think we would definitely recommend, all slightly different overall in terms of what their approach is, but they all look at vaccine research. and kind of give different sorts of approaches and really good publications to kind of read, build a level of knowledge and kind of transfer to your own development processes. So with that, and yeah, thank you all for your attention, and happy to answer any questions.
Yeah, thanks, Ian, for another great topic and presentation. And again, we thank you all for your questions and your participation. Just a reminder, again, we do our webinars the last Wednesday of every month. We'll be having another webinar on Wednesday, July 28th from 9 to 10 a.m. Pacific time. And then also our journal club. Don't forget about our journal club, which is the second Friday of every month. And our next journal club will be July 9th, and we do that at 10 a.m. Pacific time, noon Central time. So again, you can always go back and hear our presentations on YouTube, West Hour, and we hope that you have a great rest of your week, and we look forward to seeing you at our journal club. Thanks, everybody. Take care.